Okay, it's 9 o'clock here in San Diego, so we will start the webinar. Thank you very much to everybody for joining us again. The presentation today is going to be about protein structure modeling and analysis. The key topics that we'll cover are measuring when you have a protein structure, how to analyze things such as distances and angles, and how to plot sci-fi angles on Ramachandran plots, how to calculate RMSD by superimposing proteins or small molecules, how to calculate contact areas and surface areas. We'll look at the available crystallographic analysis tools that we have, including displaying neighbors and cells, and the crystallographic cell, along with electron density contouring. We'll build a homology model. We will look at an example of loop modeling, and I'll also see some very new options for predicting the effect of mutation upon binding and stability. Before we start, I just want a quick plug for a book that I co-edited with the founder of MolSoft, Ramachandran. It's devoted entirely to homology modeling, and it's a very useful book, I think, for a number of reasons, because it takes you from the very first steps of modeling, where you're trying to find a good template, and protein structure mining, through to sequence analysis, sequence alignments, force fields for homology modeling, and then different methods for modeling, including automated ones such as Swiss model, loop simulations, and how to compare protein structure, different methods for protein structure comparison, and then modeling for some challenging targets such as GPCRs, transporters, membrane proteins, and antibodies, and then it finishes up with protein-protein interaction modeling and other chapters like that. So that's available here. Okay, so in ICM, for structure analysis, we will show how to calculate RMSD, as I mentioned, contact areas, measure distances and angles, planar, dihedral, and build fully interactive Ramachandran plots, and how to superimpose proteins. So we'll start with loading ICM. I just went to the search tab and typed in the PDB of interest, which is 3BHH, which is a CAMK2 protein kinase. It's interesting because it has 4 molecules, and in 2 of them they have a ligand, and in two of them they don't. So in this example, we'll just compare the apo structure to the holo structure. So if we display the A subunit, and just for clarity, we'll color by molecule. So you see the A subunit has a ligand here, and the C subunit is in the apo form. So to compare these 2 structures, I'm just going to move the C subunit out of the object to make a separate object. So I right click, say move from object, and now we have 2 objects, 2 items here. And I'm going to superimpose these 2 C molecules. So to do that, I right click and drag over the 2 to select, or you could just select, double click in the ICM workspace, press Control and double click here. And then in the display tab, there's a button for superposition. So click there, and you see the 2 are superimposed. You can superimpose, for example, on the C terminal just by selecting the C terminal and superimposing, or you can superimpose on the N terminal, same way. Okay, but for this example, we're just going to calculate the RMSD the main difference in these two between the APO and HOLO is in the N terminal here. So to measure the RMSD difference, the RMSD here, we can do that using the options in the tools analysis RMSD here. So to do that, we need to select the first molecule we're going to compare. So we double click here, and you see that we have green crosses, which means a selection, and that's returned to a variable name called AS_graph. So you could say display CPK AS_graph, for example. Now, so in the command line, we've got a number of options. But what we're going to do is we're going to turn that variable into a selection into what we call a second selection, which is orange. So this enables us to make a second selection. So we have one selection which is orange, and the other one which will be green. So you see that the orange selection is placed into into AS2_graph here. So we have two selections, one called AS2_graph and one called AS_graph. So we need to go then to Tools, Analysis, RMSD, and you can tell ICM to superimpose the structures, but we've already done that. So we will choose the option Keep in Place because we want to see the difference as it is. So the Keep in Place option is important if you're, for example, measuring the RMSD between a ligand from a crystal structure and a ligand that you've docked. So it won't try and change the superposition. It will measure it as it is. You can change the RMSD by alignment or exact match. You can measure the RMSD of the backbone or all selected, or the C alphas, or just the heavy atom. And we'll do it as it is. Go Apply, and the RMSD is reported in the terminal window here. So that's 1.7 angstroms. Okay. So to remove the selections, we click on this red cross through the here. So we'll move on to measuring distances and angles and torsions. So display sidechain. I think we covered this in the first webinar of this series, but just to go over it again. To measure a distance, we click on the Display tab, click on the Distance button here, and you can measure the distance here. Just zoom into that region. You can change the font by Edit Font, make it larger, for example. Change the color like that, and you can see that in the ICM workspace we have a 3D label here, so we can toggle that on and off. Similar thing for angles. I click the Angle button, then choose 3 atoms, and you see that the angle is displayed here. We got a new option in the ICM workspace here. So once again, to edit that, like so. And you can also move atom labels. They have a little sort of thing you can grip in the bottom left-hand corner that you can use the middle mouse button and drag these labels to wherever you want them to be. Okay, and the other angle obviously is torsion angle, dihedral. So we can select 4 atoms, and then that will report the dihedral here. Dihedral angle here. Okay. So that's atoms. So to measure contacts between, for example, we might be interested in the contact between our ligand and the receptor. I'm going to display these and the labels. Let's display the ligand again. So we can select the ligand, go to Tools, analysis, and there's an option called contact areas. Go OK, and this displays all the residues that are in contact with this ligand. So residues that are making bigger contacts are in slightly thicker stick, and ones that are away, maybe not making so much contact, are in thinner stick. And all the information about contact areas, exposed area, and the percentage difference between the two are in this table, which are fully interactive. So if you double click, it will select the residue that you selected here. And it also reports the closest atom to that particular residue, to the selection that you made, and the distance as well. 3 points. So crystallographic tools. We have a number of different options. You can display the crystallographic neighbors. Just view the crystallographic cell. For example, here we also have biomolecule generators. So if you have one subunit of a big capsid, in this case, it will generate the whole protein. You can also read in electron density maps and contour those as well. So in this example, protein I need is 1CTG. So I display the background in white. So if we read in this crystal structure, we might be a bit confused as to how this maltose ligand actually is bound to this protein. It's right on the surface. It's sort of hanging in space. So if you took this protein and you decided to take this ligand out and redock it as is, it won't redock that way, because there is a separate chain here which is not displayed in the crystal structure as you read it from the PDB. So you need to generate the crystallographic neighbors. So to do that, we select a region that we're interested in, go to Tools, X-ray, and here are all our crystallographic options. So we can choose crystallographic neighbor and choose a radius around the selection that we've made. It's telling us the selection. These options just to increase the selection a little bit, and exactly what you'll display. We'll just press OK and we'll see what we get. So here we see that we built the neighbors. That's going to be it for the whole thing. And it generates extra molecules around the for the crystallographic neighbors, and you see that you have in the ICM workstation now have all the neighbors displayed here, which you can toggle on and off here. And then we can see that this maltose actually interacts with the other crystallographic neighbor here. So this is an important first step before you do a docking or something like that. You need to really analyze your crystal structure along with other things such as resolution and B factors and occupancy. This is just one useful tool. You can display. I'm just going to stay with this structure because I know it has a. So to display the maps, the crystallographic maps, we go to Tools again, X-ray, and there's an option called Get Electron Density Map. Click OK. This goes to the Uppsala database in Sweden and downloads that map. So you can view the map like this, but you may want to contour the map. To contour the map, you might make a selection. So we could, for example, select the residues surrounding this ligand. It's going to. So you go to right click on the ligand in the ICM workspace, choose neighbors, select 12. The type we're going to select in the same object. We don't want to exclude the source, we want to keep the source, which is the ligand. Go OK. You see we have green crosses. We can propagate that selection to all atoms in the residue and display all atoms here. And then we can contour that map by going Tools, X-ray, Contour Electron Density. Go OK. Okay. And by default, it's colored by at two different sigma levels, at 2.5 and 1. 2.5 is in blue. You can display the. You can change the sigma level yourself manually. So, for example, here we have let's undisplay that map. You can change by clicking on the plus and minus here to change the sigma level and contouring of that map. Okay. So this is also another useful tool to use before undertaking a structure-based docking or screening, because you can check whether how much of these residues the crystallographer has actually seen. And in this case, it's pretty decent. It's a 2-angstrom resolution structure. And finally, for the crystallographic section, if you want to display a crystallographic, the biomolecule, the full molecule. So here we have a crystal structure of one subunit of a virus. We can build a full biomolecule by going Tools, X-ray, Biomolecule Generator of the displayed object. Okay. I did this in practice and it was pretty quick, but we'll see how it goes with WebEx. It's making 60 units of the biomolecule. I've done it. Okay, so click here to display. We can color by molecule. And here we have the full capsid of this virus. Okay. Right. Now we move on to modeling. So the ICM has been in development for many years, probably just over 20, at least over 20 years, and it's been used in many successful modeling experiments. More recently, it's been particularly successful in GPCR modeling, and ICM did very well in the GPCR modeling competition that was arranged back in 2010, and I think there's another one. Well, there was one last year and one before that. And the results of that are published here. And now you can view the models using active ICM. The competition is arranged by UCSD and Scripps, and they use ICM to present the models and compare the models. Another interesting GPCR modeling success story was before the agonist structures of GPCRs were available. Ruben Avagayan and Seva Katic, who's at Scripps, managed to accurately predict the movement of one of the helices in agonist with an agonist bias to a GPCR, and they predicted that the helix moved out to enable the agonist to bind, and then subsequently the crystal structures of the adenosine A2A GPCR was solved, and their prediction was highly accurate. And that story is written up. The comparison with the crystal structure is written up in this paper here in Trends Pharmacological Sciences in 2011. So to build a model, you need a query sequence, obviously you're going to build, and a template from the PDB to build onto. And you can build your model, and then you need to be able to refine those models, predict problems, and also refine those models. And then also there'll be certain regions such as loops which you'll need to improve. So you need to build a loop and model those loops. And you also optimize them using a force field. We have a new force field which is described here, developed by Max Kotrov and Ruben Ovagain and Yelena from MolSoft. And we'll also show new options for mutations and binding, predicting the effect of a mutation on binding and stability. Okay. So to build a homology model, we need to get our query sequence. If we open ICM, I'm just going to take a query sequence from UniProt. We're going to build a model of this kinase. I'm going to choose it's a CAMK kinase, and if we basically went to UniProt and we get a table of hits, if you double click here, it loads the sequence of that query here in the ICM workspace. And then now we're going to do a blast search to find proteins in the PDB that have similar that would make a decent template for this query sequence. So we go to Search tab, choose Blast, and we're going to search this sequence here, KCC1b, our query, against the sequences from the PDB. So we click here, Run Search. Took a bit longer than usual. Yeah, then we get a hit list of PDB structures here. For example, they're listed in the table here with the chain number, a letter, and a code. How good the identity is, the length of the sequence. You can see the coverage on this plot here. So we can, for example, load in alignment, shown here. That's our query sequence against the hit from the blast search. So I'm going to choose one of these. These are all pretty similar, so we can choose some of these to start modeling with. Choose 1A0606. So if you go to search, PDB search, it's going to make that. So this will be our template, 1A06. And we have the query sequence here, which we're going to model. I'm just going to delete these. So we need to extract the sequence from our template structure so we can make an alignment to build a model. So we right-click on the A subunit and choose Extract Sequence. Now we have the query and the template, and we can make an alignment by selecting one, press Control, select the other, and right-click and choose Alignment, Align Sequences. or you can drag and drop onto each other. And you see the sequence alignment here. We can color the structure by the alignment. So we can see that we have pretty well conserved between the template and the query. If we change the coloring, from 2 weeks ago we showed how to do this. Maybe by constellation may highlight the differences better. So now we can, if you wanted to, you can edit. We have some gaps between the query and the template. So you can see here that there's a missing chain loop here in the template, so we need to be careful of that. So ICM will build a loop here, and we can edit the alignment if we wanted to. In this example, the alignment is pretty good, but if you wanted to edit it, you can select a region and use the cursor keys to drag your alignment along and improve the alignment. Okay, so to build the model, we go to the Homology menu. We choose the option called Build Model, and it asks for the query sequence, which it's worked out is this one, KCC1B. And the template is the A molecule of the object 1A06. Then we have a number of, and then we choose the alignment. The alignment is called ALN. So we choose the alignment here. Then we have a number of options. So this will build sort of like a raw model. So it's going to build a model, and if it finds a gap, for example here, it will build a loop. But you need to go back to that loop once you made your model and really optimize that region, because you need to make sure that the loop is correct, which we'll show in a bit. So it's going to build loops up to the length of 100 residues. At the end terminal, if there's a bit of overlap, which we have here, for example, we don't have any template, it will just build one residue at the end. You can change that preference. And the same with the C terminal. We have a lot of overlap for our query, but there's no template in this C terminal. You can allow ICM to try and improve the alignment a little bit and add gaps where needed, and there's an option to expand the gaps by 1 residue point. So these are options which you need to choose with care, because it's going to take a long time to run. You can choose. We won't choose in this example, but you can choose to minimize side chains or sample the side chains. But the most important thing is to really be aware of which part of the model you're most interested in. So if you're looking for N-terminal, if you're looking at this is a kinase, so if you're looking at for ATP binding site, or trying to model allosteric pockets in the N-terminal, you may not be interested in really bothering to really build loops and optimizing the C-terminal. So you have to just be careful about what you waste your computing time on optimizing. So in this example, we'll just build a raw model. Go OK. And it's built the model. So the template, the model we built is called KCC1B_human. You can color by object, and that will color our model structure green, and the template is yellow. So you can see ICM tried to build these very long. It's missing an activation loop here, so this region is going to be incorrect. So you need to go through your model and identify regions where the modeling is probably wrong. likewise in this region as well, we need to be careful. So you can spot these regions in the alignment where there's gaps. So once you've built your model, you can analyze the health of the model by going to Tools, Analysis, and there's an option called, Tools 3D Predict Protein Health. So we select the model, go to Tools 3D Predict Protein Health. And we obviously got a lot of problems with the model in terms of clashes, high energy problems, because we didn't optimize, we didn't do any minimization or optimization in this example. So basically this flags the energy is plotted, a normalized energy is calculated, and that's compared to statistically compared to other residues in the PDB and their preferred energy. So anything with a very high normalized energy is likely to be making a clash. So this builds a plot, and you can select in the plot. It finds a region where there's a problem. So if we displayed this residue, my guess is if we display the neighbors to this residue, there's going to be some clashes. And let's propagate to all residues in the selection using this R button. Yeah, we've obviously got major problems here with these things. So we can, the optimization would most likely flip this residue away, and likewise with this one as well. If you want to do some fixes yourself manually, most likely if you minimized it and optimized the structure, you're going to find maybe little problems which you can fix yourself. So you can select residues to sample. On the right hand side there's a residue selection button. Select one, select the other, and then you right click and choose Advanced, and then Optimize Side Chains, and then it's running the ICM in the ECEP force field. It's running the bias probability Monte Carlo simulation to try and fix this clash. You see that the lysine is moving away, and sampling base residues. In a real life situation, you'd probably want to select a larger chunk of residues in this region. And then we get a stack of energetically favorable conformations for these. Okay, so that builds the backbone of your model. You then need to obviously make the optimization and minimization. Then you need to go back to your loops and maybe remodel critical loops that are important for the task that you're undertaking. So I have an example of loop modeling. Okay, so for loop modeling, we just have an example here of this is the anthrax protective antigen, and it's an interesting example for a couple of reasons. One reason I say is that in the high resolution structure, it has a proline that is in a cis conformation, and in the low resolution structure, it's in trans. So it's unusual that this proline here. So we're just going to display this region here. Click on here. Okay, So we have a loop shown here. You see we have a proline here. Just going to see that the alignment is linked, so we can also select the. So we have the high resolution structure, which is 1 ACC, and we have the low 3.6 angstrom resolution structure 1 TZO. And we coloured the ribbon by object, and we can do the same with this stick. So we colour, click and hold on the stick, colour by object C. So if I undisplay, if you look at the low resolution structure, the green one, colour by C. You see the proline here is in trans, which makes sense, which is energetically favourable. But interestingly, the high resolution structure has that proline in cis. So what we did was to check whether this cis conformation is energetically favourable. We can use ICM to model that loop. So to model, there are two ways of doing it in ICM. They need to be ICM objects, so you right click and convert PDB. So we took in this example, we take the low resolution structure and see if we can optimize that loop to find the cis conformation, which is found in the high resolution of that proline. So we right click on the low resolution structure, convert to an ICM object, optimize hydrogens, optimize the asparagines, glutamines, optimizing the hydrogen bonding network, and go OK. We had some questions on chat, which is great. I think I might have to cover them at the end. I'll try my best to cover them at the end. So we have the protein. So to sample that loop, we have two ways of doing it. You select the region that you're interested in. So I'm just going to select the sticks. You go to mole mechanics, and then there's a sample loop option. You just get to the results. I have a ready-made simulation. But basically, we just do the ICM object. You select the residues we're interested in, 410 residue roughly here. And you go to mole mechanics, and there are some different options. So you can design a loop. You can, which we're not covering here, but you can build your own loops. We're going to sample. We have two options. One's called interactive, and the other's called high precision. The interactive will go in the distribution of ICM. We have a database of loops from the PDB, and the interactive one will run through that database looking for loops with similar distance between the start and end, and we'll build you a stack of different loops. But for high precision sampling, you can run the Monte Carlo simulation and sample the loop, and this is what we did in this example, and we have the results from that. And that will do a full optimization of surrounding side chains if you want to, but it takes a longer to run, and it's more rigorous. But this option will give you a selection of loops which maybe you can maybe optimize. You choose the best one and maybe optimize them using this option. And so we ran this simulation, and the result is that you get a stack. You see that this is the results. It's from loop 410 to residue 410 to 414. You get a stack of You can view that stack, different energetically favorable conformations like that, play the stack, or you can click here and go individually through the stack. And so we look at the first one, and we can see that, in actual fact, the cis conformation of that proline appears to be the most, seems to be energetically favorable. So we also find it when we run a very extensive loop simulation, we also find the cis conformation of that proline, which is also found in the high resolution structure. Okay, so the final thing we're going to cover in modeling is the effect of mutation. So we have two methods available. You can predict the binding free energy, which is the change in binding free energy of a protein complex upon the mutation of a single residue. And we also have an option to predict protein stability upon mutation. So to do that, so to make just to cover a couple of things, to make a mutation, rather than predicting the effect of the mutation, just to mutate a residue, you can do that by right-clicking on the residue and choose Advanced. It needs to be an icing object, so just a second. So to make a mutation, you right-click on a residue and choose Advanced, and then there's an option to mutate, and there's a list of 20 natural amino acids. Or you can right-click and choose Advanced, and we have a database of unusual amino acids that you can choose. It's also a convenient way of, for example, you can change the charge, flip the particular options based on the residue that you want to mutate. So you go to Advanced and then change here. So once you've made the mutation, you probably want to maybe do a small minimization at least, or so you right-click on the residue that you mutated, Advanced, and then there's minimize side chains, or you can do a longer simulation by optimizing the side chains. Okay. To predict the effect of a mutation, we zooming on here. You select the residue you wish to mutate, so it's methionine. Go to the mole mechanics menu, choose try mutation, and then you can select either prediction of protein binding upon mutation or protein stability. And then when you run this, you get a table of the delta G, for this example, for binding, and you can sort this table by binding. If you double click on a particular mutation, you can display that mutation. You get a separate object with that mutated residue in it. So the options, you can just you go to try mutation, protein binding, for example. You can mutate all, or you can scan the interface region, making mutations, or you can just do individually, one at a time. And that's the same also for the stability option as well. Okay, that's an overview of the modeling options. If you have any questions, feel free to email or call. The next webinar is in 2 weeks' time, and that's basically designing ligands using the 3D ligand editor. So we have ways to dock. I'll just quickly show you that. In the editor, you can dock the ligand, and you can make modifications to the ligand and optimize it. A variety of different things I can show you in a little bit. There's also coming up in May the Protein Structure and Drug Design Workshop, which is run by Ruben Abagayan, who's our founder of MolSoft at UCSD, and Max Toptrop, who's our principal scientist here. And that covers this 2-day workshop, which covers everything from graphics to sequence alignments to docking, protein-protein docking, cheminformatics, virtual screening, ligand-based screening, and structure-based screening. Yeah, so next week, it's the 2 weeks' time, it's the ligand editor. So just quickly show that. Too many selections made. And they're all about using this ligand editor tab. So you can do covalent docking, you can do multiple receptor docking, and a variety of other things. So for example, here we have a ligand, and we go to ligand editor. We can make modifications to this ligand, very quickly check the effect of that modification on the docking score and the interaction. So that will be next in 2 weeks' time. So thank you very much for joining us. And as I mentioned, if you have any questions, I'll try to answer them now. If you can write them, that would be good. Or if you need to speak to me, I can open up your mic to do that. Thank you very much. So just sorry, I'll go back in the text. So we had a question about modeling the loops. I think it's related to the loops. For modeling, there's not really a score. You just basically have an energy value which you can use to rank the. So we saw a